Hello to our friends joining us via recording. This is our second review session for the final exam. The final exam is now open. It will be open until tomorrow night, Tuesday at 11:59. So make sure you have a plan either sometime on Monday or Tuesday to take that exam. Make sure to use these review sessions to help you work on some of the maybe more challenging aspects of the exam. One of the things we're going to talk about today are the various bone disorders that we did a project based on. There were 3 bone disorders that we studied. We studied Osteogenesis Imperfecta, we studied Osteoporosis, and we studied Rickets. Before we start figuring out which statement each of these things apply to, I want to take a moment to talk about the names of a couple of these disorders. The first one on our list here is Osteogenesis Imperfecta. I hope we all can answer this question. This beginning part here, osteo. What does osteo refer to? Osteo. Yeah, anytime you see osteo, it's all about bones. So osteogenesis, osteoporosis, we had those osteocytes. All of those things, anytime I see osteo, it's all about bone. Osteogenesis, this genesis word means building. Osteogenesis imperfecta means I'm building, so genesis, building, bone, osteo, imperfecta. We can get that one, imperfect or not good. Osteogenesis imperfecta. I build my bone poorly. That's literally what its name means. Osteogenesis imperfecta. Osteoporosis, we're still talking about bones with osteo. This porosis part means my bones have pores or they have open spaces in them. So osteoporosis, bone with little holes in it. Osteogenesis imperfecta, I made my bone wrong. Rickets just doesn't mean anything, but it's an easier name to remember. Let's work our way through these statements to see if we can remember what's going on with each of these disorders. The first statement here talks about bones that are soft, and I put in parentheses here the word bendy. When I talk about bendy, we might see this in a patient as what they call bow legs, where the legs kind of bend toward the outside. So if a person's legs look kind of like this, and here I'll add a little foot at the bottom. If the legs look like that instead of straight up and down, that's what I mean by bendy legs or bowed limbs. When we talk about bone that is soft and bendy, if you will, which bone condition do we see this symptom in, or what does this describe here? Yeah, this is what's going on with Rickets. In Rickets, the bones are soft, they're bendy. When we talk about Rickets, is that a collagen problem or a calcium problem? What's going on with Rickets? Collagen problem or calcium? Yeah, so Rickets is a calcium problem. Rickets is a calcium problem. With Rickets, the issue is I don't put enough calcium into the bone when I'm building it. So don't add enough calcium. I don't add enough calcium when I'm building my bones. So Rickets is going to be a disorder that we see in children. Rickets is going to be in children because that's when I'm building my bone. The problem with Rickets is I didn't add enough calcium to that bone when I was building it. So what ends up leading to this bow-legged appearance or bendy looking bones is that I don't have enough calcium to make them strong and make them straight. So Rickets is a bone disorder where the bones are really soft. When we talk about a disorder where the bones break really easily, what did we think about this one here? Bones that break easily. Okay, so several of us are mentioning osteogenesis imperfecta. Remember, that's the one we said I built my bones wrong, right? Osteogenesis imperfecta. Yeah, and Tierney's mentioning there is actually another one too. The other one is osteoporosis. Osteoporosis. In a person who has bones that break easily, most likely what's causing those bones to break easily is because that person has osteoporosis. That's usually when we think about bones that break too easily, we're thinking about osteoporosis. It's possible, though, that someone has osteogenesis imperfecta and their bones break easily as well. Their bones would actually break even more easily than someone with osteoporosis. So osteogenesis imperfecta, bones break really, really easily. What's the deal with osteogenesis imperfecta? What's wrong in that one? Is that a calcium problem or a collagen problem? Yeah, osteogenesis imperfecta is a calcium problem, or a collagen problem, excuse me. Osteogenesis imperfecta. Yeah, so the easy person name for osteogenesis imperfecta is brittle bone disease. That's what normal people might call it, osteogenesis imperfecta. We said that the name meant bone building incorrectly. Specifically in the process of bone building, what we're doing wrong is we're building collagen that's really weak. If I build really weak collagen, my bones can't stand up to the strain or the pressure that movement puts on them. So osteogenesis imperfecta is a problem with collagen. Because my collagen is weak and not good, my bones are going to break easily. When I talk about osteoporosis, is osteoporosis a collagen problem or a calcium problem? What's causing osteoporosis? Yeah, osteoporosis is also a calcium problem, just like rickets. Also a calcium problem. But what's going on in osteoporosis? What causes us to not have enough calcium in our bones? Do we remember? Yeah, so Jacqueline mentioned the osteoclasts work a little bit more. When your osteoclasts are working more, yeah, and your osteoblasts slow down, basically what ends up happening is we steal calcium from the bones. As you get older, so that's the key here with osteoporosis, as you get older, you aren't as good at building bone tissue. We break it down faster, and also for calcium requirements, what your body needs, we're more likely to go to our bones and say, hey, you got calcium, I would love to have your calcium. So we steal calcium from the bone tissue. Literally, it leaves behind holes. So osteoporosis, bone with holes in it, the reason I have holes is because I'm stealing the calcium that was inside. So osteoporosis is a disorder where we don't have enough calcium in our bones because we stole it. We took it and we put it back into the bloodstream. Rickets is a problem with calcium because in the process of building bone tissue, we don't put enough calcium into that bone tissue. When we talk about osteogenesis imperfecta, my bones break a lot, but it's not because of calcium. It's a problem with collagen. If you remember way back from the video that we watched about the girl who had very severe osteogenesis imperfecta, her mom joked that people said, well, have you tried giving her milk? And she said, yes, we've thought of that, thank you. But that's not the problem in osteogenesis imperfecta. The problem is the proteins. It's we build collagen poorly. You can have all the calcium in the world, and that's not going to replace what collagen does, keeping your bones from breaking. So good reminder for us here, the 3 different bone disorders. The big thing, your learning objective for the bone disorders that's on that list is making sure we know what causes these disorders. So make sure we know if it's a collagen problem or a calcium problem. And this is also good to know too. Are my bones too soft or are my bones too brittle? Are they breaking too much? Good comparison between these disorders. Next thing I asked us to do with our groups was to compare the effects of different hormones when we're talking about the skeletal system. Quick reminder for us that when we talk about the skeletal system, there are two places in the body where we find calcium. So one place that we find calcium is stored inside bones. The other place in the body that we find calcium, though, is in your bloodstream. So when you're thinking about what happens with calcium balance in the body, your body is either always adding calcium to your bones because we have too much in the blood, or it's stealing calcium from the bones because we don't have enough in the blood. To help us with answering these questions here, I want to pull us back to this picture right here. From way back in the day, way back in Unit 2, when we were talking about our hormones and their effect on calcium homeostasis. Remember, homeostasis is your body wanting to keep things steady, keeping them stable. So we want to have a stable amount of calcium in the bloodstream. A number that I will not make you know is 9 to 11 milligrams. This is how much we want to have in the bloodstream. We don't always have this much in the bloodstream. Sometimes we get out of balance. Maybe we get out of balance by not having enough calcium. Maybe our diet's been particularly low in calcium lately. If our blood calcium levels go down, remember from the beginning of the, or for the middle of the semester, I guess, when my blood calcium levels go down, that activates my parathyroid glands. So my parathyroid glands are an organ that make the hormone parathyroid hormone. When we were talking about parathyroid hormone, remember one of the things we said we needed to know about this is that parathyroid hormone is released when calcium is low. Parathyroid hormone is released when calcium is low. Now, this calcium being low is calcium that's low in the bloodstream. If my blood doesn't have enough calcium, I'm going to spit out parathyroid hormone. Parathyroid hormone causes my osteoclasts, my bone breaking cells, to break down bone tissue and release the calcium that's stored in it back into the bloodstream. So when we talk about parathyroid hormone, it has effects on two things. It has effects on the blood calcium level, and it has effects on the bone calcium level. Let's start with the effect that it has on the blood calcium level. When I release parathyroid hormone, what effect is that going to have on the amount of calcium in my bloodstream? Is my blood calcium going to go up or go down when I release parathyroid hormone? Yeah, so my blood calcium goes up, If my blood calcium goes up, that calcium had to come from somewhere. It didn't just magically appear. The place that that calcium in the bloodstream came from is from my bones. Like you can see with parathyroid hormone, yeah, those osteoclasts get activated by parathyroid hormone. Their job is to break down bone tissue or to make the level of calcium in my bone tissue go down. They're breaking down bone tissue. They're, in fancy words, degrading bone matrix. So we're breaking down bone tissue. We're spitting out the calcium that's in that bone tissue to get our blood calcium to go up. So when my blood calcium is low, I spit out parathyroid hormone. That makes my osteoclasts get activated. They break down bone tissue. Bone gives away some calcium. That means that blood gets the calcium it needs. That brings my blood calcium back to normal. I'm back in balance. I asked you on the group work to predict what would happen if I had too much or too little parathyroid hormone. So let's start with too much parathyroid hormone. If I have too much parathyroid hormone, a normal amount of parathyroid hormone makes me lose some calcium from my bones. What if I have too much parathyroid hormone? What's going to happen to the amount of calcium in my bones if I do even more of what this normally does? Let's ask the question another way. What is the function of parathyroid hormone? Again, what does it actually do? What does parathyroid hormone, who does it talk to? What does it make happen in our cells? Yeah, so parathyroid hormone is a hormone that causes my osteoclasts, my bone breakers, to break down bone tissue. That's its normal job. So if I have too much parathyroid hormone, I'm sending the message to osteoclasts to work even harder. If I have too much parathyroid hormone, I'm going to really get rid of calcium in my bone tissue. If I have too much parathyroid hormone, because it talks to bone breakers and tells them to get more active, if I just keep telling them to get more active, they're just going to keep pulling and pulling and pulling calcium out of my bones. Yeah, so Ariel asked, could that lead to osteoporosis? Absolutely. This would be a way that I could make my bones get more holy more quickly, have pores where the calcium should be, because my bone breakers are just going to break down way too fast. Yeah, so overall, I'm losing calcium in my bones, which leads my bone density, how thick my bones are, to go down. If my calcium in the bones is going way down, what's happening to my calcium in the bloodstream? If it's going way down in the bones, what's happening in the bloodstream? Yeah, it's going to get too high. So way up. It's going way up. The level in my bloodstream is going to be too high. We want to get to this point, but if I keep adding more and more and more calcium, I can get too high because I'm sending this message to my bone breakers to just keep breaking things down. We want to have the right amount of parathyroid hormone that helps us get to balance. If I have too much parathyroid hormone, not only am I going to lose all the calcium in the bloodstream, or in the bones, excuse me, I'm going to lose all the calcium in the bones. It's going to end up in the bloodstream. I'm going to have way too much. We could do the opposite extreme, though. I could have way too little parathyroid hormone. If I had too little parathyroid hormone, let's start with the blood this time. The job of parathyroid hormone was to help blood calcium levels go up. What if I don't have enough parathyroid hormone? How is that going to impact my blood calcium levels? What if I can't do this half of the loop? What's gonna be the effect on my calcium? Okay, so Gloria is predicting correctly. So here we can do the bottom half of here. If I have too little parathyroid hormone, bone, well, here we'll just say it goes up. I have too little parathyroid hormone, I'm not going to activate my osteoclasts, so I'm going to keep building bone. I'm not going to be breaking any bone down. I'm going to keep building it. So if I keep building my bone and I never break it down, that means that the amount of calcium in my bloodstream is actually going to go down. I'm not going to have enough calcium in my bloodstream. Goes down. If I don't have enough parathyroid hormone, I'm not sending the message to my osteoclasts to break down bone tissue, to give me the calcium I need. If I don't tell these guys to break down bone tissue for me, and my bone builders just keep building bone, they're going to steal all of the calcium that's in the bloodstream. We're never going to put any back in. If I keep stealing all the calcium from the bloodstream, the amount that's in the bloodstream goes down, the amount that's in the bones go up. Here's a study pro tip, or a wink wink, nudge nudge. These are always opposite of each other. So if I know that the job of parathyroid hormone is to increase the activity of osteoclasts, which means I'm breaking down bone tissue, so bone calcium is going down, the opposite is always true for blood calcium. If bone calcium goes down, blood calcium goes up. If blood calcium goes down, then bone calcium goes up. They're always opposites of each other. Because remember, I only have two places in the body that we're talking about with calcium homeostasis. Either my calcium is in the bones or my calcium is in the blood. If it goes up in one place, it goes down in the other, and vice versa. So the question in the chat is if bone calcium levels go down or if they remain the same. So initially, Nicole, what would happen is they would stay stable until it's been long enough that my osteoclasts have completely stopped working. Because remember, in the background, plugging away, what we don't have on our picture here are those osteoblast cells, my bone builders. They're constantly building at least a little bit of bone tissue. If we never recycle any, which is kind of what osteoclasts do, they're the recyclers. If the recyclers are never working and the builders just keep building, eventually we are going to run out of calcium, because I'm supposed to be building bone at the same rate that I break it down. If I just keep building and I never break it down, then the amount of calcium in the bloodstream is going to go down. Does that answer the question, Nicole, or does that make sense? Okay. All right, so let's review what we've said so far before we do our second half up here. We've said that when there's not enough calcium in your bloodstream, your body spits out parathyroid hormone. Parathyroid hormone talks to our bone breakers, tells them to break down bone tissue so that calcium can go back into the bloodstream, so that it can leave bone tissue where I'm storing it, so I can put it back into the bloodstream where I need it. If I send too much of this message, that's going to make my bone breakers work too fast. They're going to lead to low bone density. I'm stealing too much calcium from the bone, and I'm probably going to have too much calcium in the bloodstream too. If I don't send this message enough, I don't break down enough bone tissue, My bones have extra calcium in them, and my bloodstream doesn't have enough calcium. It's all about balance. We want to have the right amount of calcium. We said that we release parathyroid hormone when our blood calcium is too low. When our blood calcium is too high, we're going to spit out a different hormone. The different hormone we're going to spit out is called calcitonin. Calcitonin. This is the hormone that our body releases when blood calcium is high. When blood calcium is high. We spit out a hormone called calcitonin when blood calcium is high. Does anyone happen to remember which gland in the body makes calcitonin? Who makes calcitonin? Yeah, there we go. The gland that makes calcitonin is the thyroid gland. The thyroid gland. When the thyroid gland spits out calcitonin, calcitonin, remember, also talks to osteoclasts. So we'll add this note here. Calcitonin also talks to osteoclasts. What does it tell osteoclasts when we spit out calcitonin? Does it tell them to work harder or to work less? What does calcitonin say? Yeah, calcitonin tells osteoclasts to slow down. Tells them stop doing your job. Remember, osteoclasts break down bone tissue. When we break down bone tissue, that adds calcium to the bloodstream. If I already have too much calcium in the bloodstream, I don't need to add any more. So I use calcitonin to tell my body, the way I like to remember it, is I need to put calcium in my bone tissue. Calcitonin. Bring that calcium in to the bone tissue. The best way for me to send that message is to tell my bone breakers to stop. So the bone breakers stop, which means those osteoblasts, my bone builders, they just keep doing their normal job. But because we're not adding any new calcium to the bloodstream, the blood calcium level ends up going down. We end up back at normal. So let's outline like we did before. When we talk about calcitonin, blood calcium, and bone calcium, when we talk about calcitonin, normal situation, the job of calcitonin is to help my blood calcium level go down. Calcitonin, we want calcium to go into the bones. Normal job of calcitonin, increase the calcium in bones, decrease the calcium in the bloodstream, take it into the bone tissue. If I had too much calcitonin, if I was sending this message too much, my osteoclasts would completely stop working. My bone breakers would completely stop doing their job. If bone breakers completely stop doing their job, my blood calcium level is going to get too low. So let's change my color here. If I have too much calcitonin, my osteoclasts are going to stop breaking down bone completely. They don't break down bone at all. I have way too little calcium going into my bloodstream. If I'm not breaking down bone at all, my bone calcium will go up. I am not adding any to the bloodstream. That's going down, which means that my bones are going up. They're stealing any calcium that I do have. If I have too little calcitonin, if I'm not sending this message to slow down my osteoclasts at all, my blood calcium is going to go up because my bone breakers are going to keep doing their job. But my level of calcium in the bloodstream is going to go down. It's going to go down. Or I just said that completely wrong. Let's try again. If I'm not sending enough calcitonin, if I'm not putting the brakes on my osteoclasts, they're going to keep breaking down bone tissue. When they break down bone tissue, I lose calcium in the bloodstream, or in the bones, and it goes up in the bloodstream. Calcitonin gets rid of, if I have enough calcitonin, I'm building bone. If I don't have enough calcitonin, I'm breaking bone. Yeah, so a great summary way of putting it, and this is honestly like big picture of all of this homeostasis stuff. Calcitonin and parathyroid hormone are opposites. 100% correct. I use one of them, parathyroid hormone, when I don't have enough calcium in my bloodstream. Parathyroid hormone is supposed to help me increase the amount of calcium in my bloodstream. Calcitonin is what I use when I have too much calcium in my bloodstream, when I need some of that calcium to go away. So the way that they work is opposite of each other. And the effects that they have, if I have too much or too little, has to do with what their normal job is. I know that this picture and the kind of the way we outline things is a lot to absorb. So I know we've probably got a lot of absorbing going on. But do we have any questions that we can put to words right now? Got the mind blown going on, right? So here's what I would recommend for you. Hopefully, if you wanted a picture of this, you've snapped a picture or you'll catch it on the video. So let me bounce back and go back to here. I would recommend that you try to fill this out now that we've talked about it together and you talked about it in your group. I would recommend you try to fill this out one more time to remind yourself, what does calcitonin normally do to the amount of calcium in bone? And what does it normally do to the amount of calcium in the bloodstream? Same thing. What does it normally do to parathyroid hormone, or what does parathyroid hormone do to calcium in the blood and to calcium in the bone? If you know its normal job, just think about it doing that job too well, or think about us not doing that job. Those predictions really come down to, do I remember what its normal job is? What if I'm too good at doing what's normal? What if I'm not good at doing what's normal? Shoot me an emoji. How are we feeling? I think Tyranny's emoji probably is accurate for a lot of us. Yeah, so Nicole's question asked if both of these hormones talk to osteoclasts. That is correct. Yes. Parathyroid hormone tells them to kick into gear. Calcitonin tells them to slow down. but they both talk to osteoclasts. They both talk to the bone breakers. Send you all penguin. By the way, I know we all hate it on the exams, but these kinds of questions are the kinds of things that make you do, right? I make you use the information. When you hit an application question on the exam, here's what I would recommend. First, you can do your little internal scream, right? Like, oh my gosh. Then I want you to stop and think about, or even make a note for yourself on a piece of paper, all of the normal information that you know. If I give you this question on the exam, so if I ask you, what would happen in the body if we don't have enough parathyroid hormone? That question might make you scream because you didn't memorize it. But what you did memorize and what you do know is what parathyroid hormone normally does. I promise you, every application type question that I give you, you learned the facts that you need to apply to do it. So if you hit an application question and it looks super scary, stop for a moment, take a deep breath, and figure out what information did I learn in class that Dr. Ollis is making me use to answer this question. If you can remember the normal job of parathyroid hormone, I bet you can make these predictions. I bet you can figure it out. If you can remember the normal job of calcitonin, I bet you can make a prediction to figure out what would happen if I have too much or too little. So again, when you hit an application question, take a step back, figure out what you know about it, and use the information that you know. I promise they're not as scary as you think they are. Because let's look at one of those application questions right here. This is an application question. We didn't talk about estrogen in our cycle of making bone density go up, making bone density go down. We didn't talk about estrogen. We did parathyroid hormone and calcitonin. So now we've got to figure out together what the job of estrogen is. When we talk about the effect of estrogen on bone cells, it's easiest for us to see that effect happening after menopause. So when a woman goes through menopause, she stops sending the message estrogen. So we're no longer making the message estrogen, which means whatever estrogen did, whatever estrogen made happen, is not happening anymore. As a woman ages, her bone density goes down. So let's outline what we know here. I'm going to outline it along the bottom. First, we know, number one, menopause equals no more estrogen messages. No more estrogen messages. That means that whatever estrogen does is not happening. Whatever estrogen does is not happening anymore. That's the first thing that we know. How do we feel about this fact right now? When someone goes through menopause, we're not doing what estrogen makes happen. We tracking with me so far? We don't make estrogen anymore. So the function of estrogen, not happening. We have run out of that message. The next thing that we know, number two, is that bone density goes down after menopause. Bone density goes down after menopause. So here, let's take some class information in the back of our mind to apply to this. if bone density goes down, what are the 2 ways I can make bone density go down? There's 2 ways I can do that. Okay, so we're talking about parathyroid hormones. So let's, so there Jacqueline popped up what parathyroid hormone does. Parathyroid hormone activates osteoclasts. So one way I can make bone density go down, osteoclasts get more active. That's one way I can do it. Osteoclasts get more active. What's the other way I could make your bone density go down? Yeah, there's Ariel chimed in with there. Osteoblasts get less active. Osteoblasts get less active. If my bone density, if the amount of calcium in my bone is going down, there's 2 ways that can happen. Either my bone breakers are more active, or my bone builders are less active. That's the ways I make my bone tissue lose its mass. We just talked about with parathyroid hormone, how parathyroid hormone impacts those osteoclasts and makes them more active. That's one way to decrease bone density. The other way to decrease bone density is tell my bone builders, you're good, take a vacation. That'll help my bone density go down as well. All right, so let's look at what we know from the question. Number 1, we know that whatever estrogen does is not happening any longer. The message estrogen sends is not getting sent because a woman is not making estrogen anymore. That's the first thing we know. Whatever its normal job is, we're done. The second thing we know is that bone density has gone down. So whatever estrogen does is not happening anymore, making one of these things happen. Which of these things is most likely to be happening if we stop sending a message? Do we think it's going to make somebody go faster or somebody go slower if we stop sending a message? Yeah, somebody's probably going to go slower if they're not getting a message anymore. What's happening with estrogen is estrogen normally sends a message to osteoblast cells and tells the osteoblast cells to build bone. When I stop sending the message estrogen, I stop telling my osteoblasts to build bone tissue. They don't build it anymore. The job of estrogen is actually to activate osteoblasts. I know I'm blowing your minds here because nobody voted it in the chat. Estrogen is a hormone that activates osteoblasts, that talks to bone builders. When a woman goes through menopause, we're not sending a message to bone builders to keep building bone. My bone builders stop building bone, but my bone breakers keep breaking bone like normal. When we talk about the effect of estrogen, its normal job is to keep your osteoblasts working at the right rate. If I stop telling them to work at the right rate, they're going to slow down. Best way for me to tell them to stop working at the right rate is to just stop telling them the message to keep working. When a woman stops making estrogen, she stops telling her osteoblasts to build bone tissue. Yeah, so what's happening when a woman goes through estrogen, or through menopause, excuse me, when a woman goes through menopause, essentially what happens is the osteoblasts get deactivated. They slow down. My osteoblasts have slowed down because I'm not sending them estrogen anymore. If estrogen was there, if it was working, my osteoblasts would be building bone. Since I don't tell them to build bone anymore, because I'm not giving them estrogen anymore, we are not building bone tissue. The osteoblasts slow down. I need you to help me explain this better. So Jacqueline, the correct answer is A. The correct answer is actually A. I need someone else to help me. Can we explain it in another way? Okay, so Jacqueline's, so think about this, Jacqueline. What's happening in the body to make bone density go down is the osteoblasts are not working. That is correct, that osteoblasts are not working. But here's the kicker. We're not making the message estrogen anymore. So I'm looking for what happens when I stop sending the message. I stop sending the message, my bone cells stop working. So the message told my bone cells they need to work. Let's try to outline it here. Before menopause, we have estrogen and we send the message, build bone. After menopause, no estrogen, so no message to build bone. Does this help? Or give me more questions? Okay, a couple of people say I cut out. I heard an echo. I wonder if somebody's mic flashed on. No, it's totally fine. Go for it, Ariel.
So before menopause, it activates the osteoblast, and after menopause, it deactivates the osteoblast. That's what I'm trying to figure out.
So it's not that the function of estrogen changes. The job of estrogen is always to tell osteoblasts to build bone. Mm-hmm. When a woman goes through menopause, she stops releasing estrogen. She stops talking to her osteoblasts to tell them to build bone. It's just like, I think somebody put in the chat, when we were talking about calcitonin and parathyroid hormone, it's not that what calcitonin or parathyroid hormone says, what their message is any different. It's just whether you spit it out or not. The message that estrogen always sends is it tells osteoblasts, you need to build bone tissue. When a woman goes through menopause, she stops telling her osteoblasts to build bone tissue. Mm-hmm. If we had her do estrogen supplements, estrogen would still go and tell her osteoblasts to build more bone. The problem is she's not spitting out estrogen anymore. She's not sending the message to her osteoblasts to build bone. Since she's not telling the osteoblasts to build bone, they're not building bone anymore. So the message that estrogen sends The reason that bone mass stays higher before menopause is because estrogen is telling my osteoblasts, keep going, keep doing your job. When I stop sending that message, my osteoblasts stop working. When they stop working, my bone density goes down. Yes, so Jacqueline put it another way. In basically what this question is asking is right here. Predict the effect of estrogen on bone cells. We want to know what's the normal job of this hormone. What's the normal job of this hormone? Normally, this hormone is telling osteoblasts, and always, to be fair, this hormone, estrogen, always tells osteoblasts that they need to build bone tissue. If I stop sending estrogen, then I'm no longer going to build bone tissue. Yeah, so Jacqueline, and I think a lot of people are having the same thought about it. Yep. I wasn't asking you what happens after menopause. We were 100% right about what's happening, right? My bone breakers are working harder than my bone builders. But that's not the question. The question is, what is estrogen's job? What does estrogen do? Estrogen tells my bone builders to build bone. If I stop talking to my bone builders, they're not going to build bone. So that's going to look like the osteoblasts aren't working. That's going to look like the osteoclasts are working harder. And that's correct, but that's not what the question asked. So make sure, like we're saying in the chat, make sure to read the questions carefully, especially those application questions that we all hate, right? We got to make sure we read them slowly and carefully. What I wanted us to do with this was to figure out what the job of estrogen is. The job of estrogen, when I'm releasing it, when it's doing its normal job, is to tell my osteoblasts to build bone. When I lose that message to build bone, my bone density goes down. My osteoblast after menopause, so if the question was, here, let's put some new text. What happens during menopause? If this was my question, we could say that those osteoblasts slow down, and we could say that the osteoclasts speed up. If this was my question, but this was not my question, right? So read the question slowly, take your time with it, make sure you're answering the question I'm actually asking you, not the question you think it might be. Yeah, Ariel says we got to be detectives during the exam, right? Read slowly, see what it says. We mentioned this too. You can always read questions out loud. Proctorio is not going to kick you out of your exam if you want to read a question out loud. It's not going to kick you out. So read it out loud if that helps you to understand it better. Yeah, Hannah says she doesn't want to sleep on these questions. What questions do we still have about this? Any other questions about this? Are we all weeping on the inside now? Yeah, Gloria is looking a little queasy. So Jacqueline asked about the questions on the exam. I believe that it has 60 questions, some of them with multiple different parts on them. So I think it's something like 80 points across 60 questions. So expect about 80 facts, basically, I guess. I don't know how to describe it better, but you know how it is on the homework assignments where they have multiple parts. So there are some questions with multiple parts. Briefly, I'll mention this for you all. This is in your lesson number 8 outline. What page is this picture from? Anyone happen to have number 8 out? Okay, page 11. So just want to mention for you, we just talked about this right here. We talked about how when a person gets older, notice right here, my bone builders, my osteoblasts, their rate of activity is going down because we're not getting, as we age, as this woman has gone through menopause, we're not sending estrogen messages to our osteoblasts to stay alive and stay active. So they're not building bone tissue. And the longer we go not building bone tissue, the lower the level of calcium is in our diet and in our bloodstream, the more those osteoclast cells get activated. Both of these two things lead to bone density being much lower and making it much more likely for our bones to break. So osteoporosis, the risks shoot way up in a woman after she goes through menopause. The reason it shoots way up is because we no longer have estrogen to activate those osteoblasts. So just to tie that into lesson number 8, I'll mention that. Okay, I'm looking through what I have pulled content for the rest of our session today. Here's the options. Endochondral ossification, so the process of bone building, the cross-bridge cycle, so how muscle contraction works, or the blood-brain barrier, or last thing was the molecules in the skin. Where do we want to go first? Skin, blood-brain barrier, cross-bridge cycle, bone building. Okay, I've got at least one vote for everything. All of the above. Okay, well let's see. I'm going to do bone building, then I will do skin. I think we'll have enough time to at least briefly talk about the blood-brain barrier. Gloria, I know you asked about the cross-bridge cycle. Are you available tonight at 5 o'clock? Yes, okay. So I want to make sure I have plenty of time to cover that with you. We will plan tonight at 5 o'clock. One of the things I will for sure talk about is the cross-bridge cycle. So that is on the docket for tonight at 5, cross-bridge cycle. We'll make sure we talk about that. Let's talk about endochondral ossification. Endochondral ossification. Remember way back from this lesson, endochondral ossification. Endo meant inside. What did chondral mean? What did we say? Anytime we see chondro, what should we be thinking about? Yep, we should be thinking about cartilage. Endochondral ossification. The process, ossification, by the way, is bone building. So inside cartilage, bone building. Endochondral ossification. This is the way that we build almost all of the bones in the body, except for one group of bones. What was the kind of bones that we built using a different method? Does anyone remember? It was a shape classification. Yeah, it's those flat bones. So flat bones, like the bones of your skull, like your sternum and your ribs, we build them a different way, but pretty much all the other bones in the body we're going to build this way using the process called endochondral ossification. So let's talk about how this process works. Big picture, what you should keep in mind with endochondral ossification is that the reason I build my bone tissue, it all comes down to nutrients. If I want to build good bone tissue, I need to have blood supply. I've got to have oxygen, I've got to have glucose. If I don't have that, I haven't built good bone tissue, or at least not yet. So here's how this process works. Very early in utero, we start to develop a hyaline cartilage model, that's shaped like the long bones in the body. As that model reaches a certain size, it builds this thing called the bone collar. The bone collar. Now, when we think about the bone collar on our cartilage model, this is a thin layer of bone that we find on an area called the diaphysis. That's a word to remind yourself of, the diaphysis. That was a lab word that meant the shaft or the long middle part of the bone. We build this little thing of bone tissue on the very outside of this cartilage model. I like to joke that the cartilage model gets so excited. It says, I built some bone tissue. Look at me. I'm so special. Except the cartilage that lives on the inside of the model says, dude, you just cut off my oxygen and my blood supply. So in the middle of the hyaline cartilage model, we start to calcify, or basically kill off my cartilage cells that live in the model. They send out an SOS message, we're dying. So in comes something called a nutrient artery. So a nutrient artery, just like its name says, brings in oxygen and nutrients. It passes through the bone collar into the diaphysis of the bone. It forms what's called the primary ossification center. The primary ossification center. This is where I first start actually building bone tissue. So before this, I started to try to grab calcium and build pretend bone. No good. I bring in my nutrient artery and I start to build bony tissue here in the diaphysis, the primary ossification center. Bony tissue continues to get built through the diaphysis of the bone. This continues all the way up till birth, and even after birth, we're building off of our primary ossification center here. Right around the time of birth, or shortly thereafter, we send blood vessels into the parts of the bone called the epiphyses. The epiphyses, that's the ends of the bone. I send blood vessels into the ends of the bone, and they start to build new bone tissue in the ends, in the epiphyses. Those areas where I build bone tissue in the ends are called the secondary ossification centers. So important compare and contrast for us. The secondary ossification centers are found in the epiphyses, in the ends of the bone. The primary ossification centers are found in the diaphysis, in the shaft of the bone. I've just got one of those because it goes into the middle. I've got one at each end of these secondary ossification centers found in those epiphyses. A couple of places where I don't do endochondral ossification right away or for a very long time. I never do endochondral ossification in what we call the articular cartilage. That's the cartilage I find at the joint at the end of the bones. So this will always stay cartilage tissue. We also have that place that we talked a lot about called the epiphyseal plate. What happens at the epiphyseal plate, or why do I care about the epiphyseal plate? Why is it important? Yeah, exactly. Sometimes we call the epiphyseal plate the growth plate. This is the part in all of your long bones that keeps growing until about ages 18 to 21. When we hit about 18 to 21, we stop growing because our epiphyseal plate turns into what? What do we call it after we're done growing? Yeah, we call it the line, right? The epiphyseal line. So again, somewhere around ages 18 to 21, that epiphyseal plate that was cartilage turns into bony tissue called the epiphyseal line. So when you're studying, one of the questions that it asks about is the importance or the significance of the epiphyseal plate. This is what helps you to keep growing taller. When this closes, you're done growing taller. It typically is going to close around ages 18 to 21. Questions about endochondral ossification or about the epiphyseal plate? We're feeling pretty good about this one. Perfect. Mushu's back. I like it. All right, let's talk about the skin. When we talk about the skin, there were 4 molecules or 4 compounds that I told you needed to know about. One of them was keratin, one of them was melanin, one of them was glycolipids. So let's work together in the chat to remind ourselves about these compounds. Let's start with melanin. When we think about melanin, what are some of the most important things you remember about melanin? What is it? What does it do? Who makes it? What are some of these ideas about melanin that we can think of? Okay, perfect. So the most important function of melanin, it protects from UV radiation. Melanin is also what gives us skin color, right? One of the big contributors to skin color. Melanin is made by, perfect, I see that in there, made by cells called melanocytes. Melanocytes make it. When we talk about melanocytes, remember that everyone has the same number, different activity is what makes us have different skin tones. We all have the same number of melanocytes. Some of us, our melanocytes are more active, so they make more melanin, leading to a darker skin color. Some of us, our melanocytes are less active, so we'd have a lighter skin color. The big picture job of melanin is protect us from UV radiation. Yeah, so Christina made an important last tie-in that we'll add here. Melanin relates to albinism. What's happening in albinism? We can remember what's going on in an individual with albinism related to melanin. Yeah, so albinism, remember when we looked at the pictures, your skin is very light or it's even white in appearance. When someone has albinism, their melanocytes are literally not capable of producing melanin. They have a mutation in their DNA. So when they go to build the melanin protein, they can't do it. So melanin is what we see. Let me pull up my pointer here. All of these little black spots or these brown spots that you see here, these are little melanin granules. Their job is to absorb any UV radiation that comes from outside the body that's trying to come in and bump into our cells. UV radiation is problematic because it can change your DNA. And if we change our DNA, we might develop what? What did we talk about developing because of UV radiation? Yeah, UV radiation can lead to skin cancer. Skin cancer is when UV rays went down, they damaged your DNA, especially when they damaged the DNA in the position in one of your genes that controls how often your cell divides. If I damage that and my cell just starts dividing out of control, I develop skin cancer. So the job of melanin, big picture, is to help you prevent skin cancer. It's my pigment protein in the skin. When we talk about glycolipids, glycolipids also protect us, but they protect us from something else, or they have a very different job from melanin. Does anyone remember what glycolipids do for a cell? Glycolipids? Yeah, so glycolipids waterproof the skin. Absolutely, they waterproof the skin. Glycolipids are able to waterproof the skin because of the second half of their name. It's the lipid part of them that helps us to waterproof the skin. Are glycolipids made by cells that are found everywhere in the skin, or just in some places in the skin? Who makes glycolipids? Do we remember? It's been a minute, right? Yeah, so Gloria is right. Oh, we'll tie together what Christina said and Gloria said. The type of cells that make glycolipids are called keratinocytes. Keratinocytes make glycolipids, but not all keratinocytes make glycolipids. We have to be in what's called stratum granulosum or above for us to see these glycolipids. Remember from lab that stratum granulosum was the layer of the skin, it's this layer right here, where we see the dark spots inside of the cells. So these dark spots that I see inside of the cells were called, here's our other word for them, lamellar granules. Lamellar granules. Stratum granulosum, we have these little lamellar granules that are the dark spots made of glycolipids. These glycolipids repel water that comes from the outside. We talked about, though, how they also repel water that comes from the inside. Why are glycolipids bad for these cells? What happens when they cover themselves in glycolipids? Does anyone remember? Yeah, so we don't let anything go through. Yeah, and the problem is what I'm not letting go through is important stuff that dissolves in water. Stuff like glucose, cell's favorite food. So from this point up, from stratum granulosum up, my skin is dead because these cells make a waterproof barrier. So no water comes from the outside. That's good for you. But at the same time, no water comes from the inside. The water, the interstitial fluid, that would have the nutrients in it. So glycolipids waterproof the skin, but they also kill the keratinocytes. They also kill the keratinocytes. Keep you waterproof, but also make the outer layers of your skin dead. Hey, keratinocytes get their name because they're full of keratin. What are some of the things we remember about keratin? Do we remember anything about keratin? I'll put a note here that it's made by keratinocytes. That's how they got their name, right? Yeah, so keratin's big job is to make skin tough. Absolutely. Keratin is what makes your skin really tough. We have a lot more keratin in places like your fingernails, your toenails. That's what makes them even stronger. Keratin also is used to build something, a type of cell junction that connects cells to each other. Do we remember what that one was called? It actually wasn't the tight junctions. Yeah, it was actually those weird ones called desmosomes. That would be an underline highlight star reminder, because we talked about desmosomes and tight junctions right next to each other, but it was only those things called desmosomes that keratin is involved in building. So keratin inside the cells, it fills up all of these cells here. That's why they're called keratinocytes. We use it as anchors for those desmosomes, which were the kind of junction that help us to hold together cells when they get pulled in many different directions. So keratin makes your skin tough, helps it to resist pulling when we go in lots of different directions. All the cells of the skin, all those keratinocytes make keratin. Only melanocytes make melanin to protect us from UV radiation. All of my keratinocytes, once they hit stratum granulosum or they go up higher, they're all going to start making glycolipids. Remember, I kind of told you with glycolipids, it's kind of like your midlife crisis if you're a keratinocyte. When I get this far away from stratum basal, I freak out and decide that what I should do instead of buying a convertible is I should start making glycolipids. So my midlife crisis, spit out glycolipids, and then I work my way up and off. To Tierney's question, no, glycolipids don't make up tight junctions, but glycolipids have the same job as tight junctions. So the purpose of tight junctions, I'll add a note about them here on our slide here. Tight junctions use plasma membranes together. So yeah, so tight junctions help us to waterproof. They make sure that water can't sneak between the cells, because literally neighboring cells, their membranes are fused to each other. So they do the same job as glycolipids, but their glycolipids are a fatty substance that I spit out on the outside of the cell. These are proteins that we use to attach the membranes together so that stuff can't sneak through. So the two ways that I attach cells to each other in the skin, we use those tight junctions that make the membranes fuse together, and then the desmosomes that resist pulling. And remember, desmosomes made out of keratin. Desmosomes made out of keratin. Any other skin questions? Yeah, totally fine. Ariel says she's soaking it in. I feel you. So let's do this. Our last topic for today. Let's chat about the blood-brain barrier. Blood-brain barrier. Here is my first reminder, or my first note for you when you're thinking about the blood-brain barrier. Remember that big picture, the blood-brain barrier is just made out of two plasma membranes, essentially, that separate a blood vessel from the brain tissue. I remind us of that to say all the stuff that we learned in unit number 1 about what can and cannot cross the plasma membrane, that's going to be exactly the same for what can and cannot cross the blood-brain barrier. So the blood-brain barrier is made of two layers of cells. We've got the cells that are inside a blood vessel. Their name is endothelial cells. And then we've got cells on the outside of a blood vessel. I don't think I made you know their name for the final exam, but a reminder that the ones outside the cells are called, or outside the blood vessels, are called astrocytes. So I've got two plasma membranes, one of them from the cells inside a blood vessel, one of them from the cells on the outside of a blood vessel. For something to be able to go across the blood-brain barrier, does it need to match the inside or the outside of the plasma membrane of a cell? Who can remember? Which part of a cell membrane decides what passes through? The outside or the inside? Let's do a little sketching here. I've got like a cell donut going on over here. I just put some icing on it. Yeah, so it's the inside part of the membrane, not the outside part. Remember that this middle part in here is called the tails, right? And the outer part and this very inner part, inner ring here, these are called the heads. If I want to pass straight through, straight in, or straight out of a cell, I have to match the lipid tails that are in the very middle. That's how you cross into and out of a cell. If I want to cross the blood-brain barrier, I have to cross 1 and 2 plasma membranes. So it's all those words, like Ariel put in the chat for us, all the words of stuff that we said could sneak straight through the plasma membrane. That's the same kind of stuff that can sneak straight across the blood-brain barrier. So if you are oxygen or carbon dioxide, which are two gases that are non-polar, you can pass straight through the blood-brain barrier, straight across, because you match the lipid tails that make it up. If you are fat soluble or lipid soluble, again, you match those tails. You can go straight across. If you're lipid soluble, you are also hydrophobic. You're afraid of water. You would not be absorbed in water. You are nonpolar. So here, let's make our list here. We said that the tails are nonpolar. We said that they are hydrophobic. We said that they are lipid soluble. Fat soluble is the same thing, like we see in our picture here. So when you're thinking about what can and cannot get across the membrane, anything that, or across the barrier, anything that would sneak in and out of a cell, we're good. If we want to do something like glucose, that's polar, I'm going to have to do transport. I'm going to have to use a protein channel to get it across the membrane because it doesn't match those lipid tails. So glucose is polar. Put a little note by it. Glucose is polar. Because glucose is polar, I got to use a transport protein to help me do that. Same thing here with my amino acids. Several of them are polar. Several of them have a charge. If I'm going to get them across the plasma membrane, I've got to do transport. I've got to use a protein channel to do that. You can see it here too. We've got those ions floating around in the bloodstream, positive and negative charges. Again, if I wanted these to get in and out, I would have to use a transport protein to do that. As usual, when you see questions that ask you about what can and cannot cross the blood-brain barrier, it's going to be an application question. You don't care about what I put in front of my description of the chemistry of things. If I tell you that something is non-polar, it doesn't matter if I'm talking about a medication or a food or a chemical. It doesn't matter what I say it is. If I say it's non-polar, that thing can cross the plasma membrane. You don't care if I'm talking about a medication or a chemical or a food. If I tell you it's polar, that's not going to cross the plasma membrane. So these questions will look tricky when talking about the blood-brain barrier, because I'm going to give you the name of chemicals and medications and all that jazz, but they're really not tricky. The blood-brain barrier is a block that keeps everything out that's not nonpolar, that's not hydrophobic, that's not lipid soluble, or the other thing that we had in the chat that we didn't really mention here, small things might also be able to cross the blood-brain barrier. So very small things would be the other thing that might sneak through. So for the blood-brain barrier questions, again, it's just an application of the stuff that can pass through the cell. If you can get through the cell membrane, you can get through the blood-brain barrier. If the membrane says no, then the blood-brain barrier is going to say the same thing. You shall not pass. I'm probably going to call that good for content today. I am going to tonight plan to talk about the cross-bridge cycle. What other topics would we like to talk about tonight? Any other particular ones? Yeah, so we'll talk about the cross-bridge cycle. I'm looking through what I had pulled. We can do some practice with how to do the processes of transcription and translation if we're interested. I think I had some questions about prions possibly that people had asked. Yeah, go ahead, Miriam.
As a review thing, is it possible for you to open those homework assignments that we already done for each lesson?
So you should be able to do that. The other thing I'll mention though is I did create some final exam review assignments that only cover the stuff that is included on the exam. So there is content from each of the units that I'm not asking you to relearn for the final exam. So I kind of made a smaller batch of questions for you on those review assignments. So there is a folder that has review assignments in it that you can go through and do. They're not graded, so in the sense that it's not going to affect your grade. You could go through and try those. I think all of the other assignments, if you wanted to redo them, you can. It's not going to change your grade in the grade book, but if there's a particular one you wanted to go back to, I believe you should be able to do that on most of them.
Okay, so just to make sure that I got this correct, so you said the two assignments that you put up for review for the final exam, those are the assignments that you have the topics that is mainly covered in the final exam. Did I get that correct?
Yes, yeah. So I went through and took all of the question pools that your homework assignments were pulling from, and I just pulled the questions that relate to the stuff on the learning objective list for the final exam.
Oh, awesome. Thank you so much for doing that.
Yeah. And you'll notice, I know I have some friends here who've done it multiple times. You get different kinds of questions each time. So the more times you can go through and submit it, the more variety you'll see. But you shouldn't see any topics in that review assignment that are not included on the exam. I think I pulled out those ones.
Thank you so much.
Yeah, you're welcome. All right, well, I will plan to cover the cross-bridge cycle. We can compare ions and proteins. So probably I'll throw carbs into the mix too in terms of functions for tonight. Yeah, Gloria is asking about critical thinking questions. Yeah, I guess that's why there's good motivation to come to class, right? To do critical thinking questions together. I don't have a lot of critical thinking questions to put out there because I have to brainstorm them up. So unfortunately not a lot of those, but you can always go back to and look at the very beginning of each of the recordings from this semester, because we often in our group work at least do application type stuff. So you can always check those out and see if there's any more practice like that to go through, because I never, on the group work that we do at the beginning, we never do it exactly like it is in the notes on purpose. So maybe give that a try. That's as close to extra critical thinking practice as I can get for you. All right, well, I have a list of some stuff that I'm planning to cover tonight at 5. If you think of other things, feel free to email me. Otherwise, I hope to see several of you tonight at 5. If you have already taken the exam by then, that's totally great and awesome. I hope to see you tomorrow morning at 9:30 for the Salty Banana Challenge. So get those bananas, bring that salt. I look forward to trying it with you all tomorrow. So see some of you tonight at 5. Otherwise, good luck on the exam, and I will see you tomorrow.